Hello everyone, my name is Mohit Kumar from group 317. Today my topic is about the composition and the primary structure of protein and nucleic acid and some electrolytes. There are the four levels of organization of protein. The first is primary structure of protein, secondary structure, and then tertiary and then quaternary structure of protein. The primary structure of protein, in which the amino acids are arranged in linear sequence. As we can see, the amino acids are arranged in linear sequence and they are connected by the peptide bond. And in secondary structure there is a folding of, we can see there is a folding of polypeptide chain of that primary structure. And in tertiary structure there is a folding of secondary structure and they form the three domain. We can see in 3D the tertiary structure. And in quaternary structure there is a folding of two or more than two polypeptide chain. There is a four level of organization. In primary, the linear sequence of amino acids, and in secondary there is a folding of the primary structure. In tertiary there is a folding of secondary, and in quaternary there is a folding of tertiary and secondary. The first is primary structure. The primary structure depend upon the number of amino acids. We can see there is a number of amino acids which are connected by the peptide bond. So the primary structure depend on the number of amino acids and the sequence of amino acids. In primary structure, we can see the amino acids are connected by the peptide bond. There is a diagram where that we can see N terminal or C terminal, and the peptide bond we can see there is a R1, R2, R3, R4. These are the alanine, guanine, any amino acid we can see. The example of primary structure is insulin. Insulin have two chain, A chain and B chain. Both chain are connected by C peptide. In immature insulin, the C peptide is breakdown and form only at mature insulin only two chain, A chain or B chain. The second is about secondary structure. Secondary structure, we can see alpha helix, beta plated sheet, turn, blends and loop. The first is alpha helix, which is secondary structure of protein, which is most common and spiral. The bond in alpha helix are hydrogen bond and the interaction are hydrophobic interaction, and there will be two forces in alpha helix structure, which is electrostatic forces and van der Waals forces. This is a structure of alpha helix structure which is spiral in shape, and this is stabilized by hydrogen bond. The example of alpha helix structure is myoglobin and hemoglobin. In hemoglobin and myoglobin chain, alpha helix structure is present. Second is beta plated sheet, which is also a secondary structure of protein, which are further divided into two parts, the palar and antipalar. This is arranged in zigzag manner. We can see this nitrogen and this nitrogen is antipalar, this carbon and this carbon is antipalar. In palar, antipalar, and we can see this nitrogen are palar, this carbon is palar. The example of beta plated sheet is silk protein carbonic anhydrase. Carbonic anhydrase is present in respiratory CO2 O2 transport. Third is the tertiary structure. In tertiary structure, we can see the folding of secondary structure to form the three domain. The tertiary structure is in myoglobin, and myoglobin is present in skeletal muscles. The fourth and at last of protein is quaternary structure, which is formed by two or more polypeptide chain. This famous example is hemoglobin. Hemoglobin have two alpha chain and two beta chain. We can see the two alpha chain and two beta chain, and in that chain there is a ferrous group present in hemoglobin. My second topic is about nucleic acid. Nucleic acids. What is nucleic acid? These are the polymer of nucleotide. And nucleotide are the nitrogen base, pentose sugar and the phosphate group. There are the two type of nitrogenous base, the purine and the pyrimidine. Purine contain adenine and guanine. These are the structure of purine and the guanine. Pyrimidine contain three structures, that is cytosine, uracil and thymine. Uracil present in RNA, thymine present in DNA. These are the structure of cytosine, uracil and the thymine. The next is sugar pentose. There is a two type of sugar, ribose sugar and deoxyribose sugar. Ribose sugar present in RNA, whereas deoxyribose sugar present in DNA. We can see the structure of ribose sugar and deoxyribose sugar are same, but there is a little difference. In deoxyribose, the second OH will be removed. The third is about phosphate group that attach to that ribose sugar and the nitrogenous base and form the nucleotide. We can see this is the structure of guanine, this is the phosphate group attached, and this is the ribose sugar and form the deoxyguanine monophosphate, which is base sugar phosphate, which form the nucleotide. If the base is adenine and the nucleoside is adenosine, the nucleotide will be adenylate acid. If the base is guanine and the nucleoside is guanosine, then the nucleotide is guanylic acid, like uracil. If the base is uracil and the nucleoside is uridine and the nucleotide is uridylic acid. The difference is about that thymine present in DNA whereas the uracil present in RNA. There is the chemical structure of DNA and RNA. DNA model was discovered by Watson and Crick in 1953, that is double helix model. There is the rule called Chargaff rule. Chargaff rule, according to Chargaff rule, the amount of purine is equal to the amount of pyrimidine. This is the structure of double helix structure of DNA. In DNA we can see the adenine form 2 bond with thymine, whereas the guanine form 3 bond with cytosine. These bond are the hydrogen bond. We can see the minor groove and major groove. Major groove is for regulating protein. This is the 5 prime to 3 prime. This is the 3 prime to 5 prime, which is anti-parallel the structure of DNA. This is the right-handed helix structures. My third topic is electrolyte. What is electrolyte biophysics? Electrolyte biophysics is a study of electrical property of electrolyte, which are substances that conduct electricity in the body. The main function of electrolyte is they play the important role in maintaining fluid balance, regulating pH value, enabling nerve and muscle function, and hydrating the body. Some electrolytes are sodium, potassium, calcium, magnesium, chloride, phosphate, and bicarbonate. These electrolytes are also used in the sodium-potassium pump and channels. There is some biophysical property of electrolytes, that is ionization, conductivity, diffusion, and osmosis. The titration of weak acids of a strong base by NaOH. These are some reactions which is called alpha, that is the degree of dissociation. This degree is mainly for the weak acid and weak base. For strong acid and strong base there is no degree of dissociation alpha, which is only for weak acid and weak base. So alpha, this is a formula of degree of dissociation. What is pK? pK is meaning the concentration or the strength of pH. It depends the strength of weak acids, the weak base, that is the pK. So here is some amino acids, their pK1 and pK2 value. For example, glycine, the pK1 value is 2.35, pK2 is 9.72. We can see the pK2 value is always greater than pK1 value. The second is the valine, and the pK1 value of valine is 2.32, and we can see 9.62 is the pK2 value. As we see the pK2 value always greater than pK1 if there is a pK3 value which is greater than pK2 value. Some electrolyte biophysics important. Electrolyte transport. The movement of electrolyte across cell membrane including active or passive transport. In passive there is along the gradient, and active against the concentration. In active we need the energy ATP, whereas in passive we cannot need energy, we cannot need ATP. Second is ion channel, like sodium potassium channel, sodium potassium pump. These are some channels or pumps. Next is electrolyte imbalance. If there is an abnormal level of electrolyte in body which can lead to various health disorders such as dehydration, rehydration, that type. Electrolyte regulation, the body mechanism for maintaining optimum electrolyte level by taking the fluids if we are suffering from dehydration. Electrolyte therapy in medicine, the use of electrolyte as a treatment for various medical conditions including electrolyte deficiency imbalances. Thank you very much.